Okay, everybody. Hi. And looks like we have a good amount of people here, and welcome to our session number 3, case studies number 2, retroactive data sharing, and our lysosomal storage disease data sharing workshop. My name is Kara Wiseman, and my co-host, Krista Kazaz, will be joining us in a little bit. She will have a special presentation for you. I am the director of the Program of Excellence in MPS and Lysosomal Storage Diseases, and I'm very grateful to share this third session with you both, representing both the Orphan Disease Center and the Critical Path for Lysosomal Storage Diseases at the C-Path Institute. So just a little overview here. So as you may recall, the workshop goals here are to produce materials that can be used by sponsors and patient advocates and patient advocacy organizations and other patients that will be going into a study, either a natural history study or a registry, and helping them to share data and guide them on how data can be shared from this work. So in the first 2 sessions, which I'll go over in a little bit, we both set the scene for the studies that we were going to be talking about. And then we held a proactive data sharing case study, which talked about how sponsors approach future data sharing before the study begins. And as this shows here, we're together talking about retroactive data sharing, and we'll talk more about that in just a minute, about how sponsors approach data sharing once a study has already begun or has closed. So if you have any feedback, we definitely encourage this. We're going to be using a lot of it for writing these papers. And so we encourage you to go to this feedback form or QR code here. You can find this on our website, and you can also find this through that QR code. And here we will be taking that information back from you and being able to write this material. So please, we also would encourage using additional case studies that maybe were not highlighted in these presentations online here, but instead will be presented in a written form. That information will be extremely valuable for us as well as we put this material together. So please give us feedback at any time, and please have more case studies if you'd like to share them. So for session 3, as you can see here, we have a really exciting group of very diverse speakers that we're going to be talking through today. And we're going to be looking at retroactive data sharing from a bunch of different angles here. So the first presentation that we're going to have is going to be talking about the BDSRA Foundation and their work with Batten's disease. Noah is going to be speaking from a community and database manager position and talking about the work they're doing setting up for this retroactive data sharing that they will be doing. Then we'll be having Krista actually present, which is why she's not presenting now, to talk about her work with CPLD in more depth. I'm excited for her to get into this material and to show you guys the work that she's been doing with a bunch of different sponsors here to encourage them to share data. And then we'll have Heidi talk about the Rare Disease Cures Accelerator, which is the platform that C-Path uses, and it's being used in a lot of the different examples we're talking about over the last couple of months. So it's a great platform. She'll give you examples of that work. And then we're going to really get into the meat of a study with Janine Jarnes from the University of Minnesota, talking about the natural history study of gangliosidosis, which she's been working on for many years. Then we'll go into our Q&A with our panel, and then we'll have future discussions. So we do ask that to put information and questions, please use the Q&A function below. You'll see, and you will also be able to have the option to speak using your own voice rather than just the Q&A when the Q&A time starts, which will approximately be around noon, give or take. So please feel free to either put the questions as you see fit as the presentation continues, or please feel free to speak up when the time comes. So just as a quick summary of what we've learned so far. So in our overview, we learned that data sharing is much greater than siloing or hoarding that data, but I think we're all on the same page there. And that really the consent language determines the study's data sharing ability. But there are also other aspects which we will get to today that you need to do to make sure you're able to share and have the institution be supportive of it. We also learned about biomarker identification, and Krista gave us a lot of information on that, talking about how we understand disease progression, identification of patients for early intervention, and the potential for accelerated approval. And we'll get into more of this as we move on. And then in session 2, just a couple of highlights here. We learned about platforms for rare diseases and how that can help to study and coordinate streamlining data sharing efforts, and how natural history studies goals are often to include identifying pre-symptomatic children so we can intervene therapeutically appropriately. But there are some challenges to these natural history studies and registries, including defining subgroups, especially when it's identified at birth. With our natural history study data harmonization and curation aims, they want to bring the same rigor as a clinical trial so they can be used as such. There are other needs and other usage for this data as well, but in a lot of these cases, that is what the goal is. Like we talked about earlier, it's a lot of consent-based data sharing. It's important for that consent to indicate what we're going to need for IRB and rec approval documentation and how that works for getting the material shared after. Also, we want to design these studies with data sharing from day one or earlier, but we're going to learn a little bit more today about how it looks when you're not thinking in that way, but you are excited about sharing data and you're going to do it after the study has already started. And there are differences between academic and nonprofit data sharing that we learned about. And then we learned about data samples that can be collected and shared with those MTA and IRB approvals and how that works. So a lot of this will all be included in our final material that will be presented in the next couple months and years. Hopefully, lots of data will come out of this. But at this time, this is where the information that we have so far, and we look forward for this to grow. So as we learned about the Orphan Disease Center and CPLD previously, I'm not going to get into this a lot, but just to kind of level set for everybody, we really both work in rare disease research, and we do patient advocacy as well. And research and advocacy in action is the way that we look at this. So really quickly, the Orphan Disease Center works on the research side where we do patient-focused, appropriate, and timely and barrier-free therapeutic development. And we also not only invest in the research side, but we also invest in the natural history studies and registries associated with these areas. So this is what brings us to the table today. And when you look at the rare drug development regulatory overview, we are all familiar with this process. And we learned also that when you are going through the drug development timeline, it is important to have these registries and natural history studies because of this information can accelerate the process in many different ways, and we will touch on more of this today as well. Okay, so when we're looking at retroactive data sharing, these are a couple of things that might come into play today. Some of the areas we might hear about and some we might expand on here, whether that be modifying consent language, having a reconsenting process, the legal agreements that are necessary for that work to happen, the regulatory involvement that might occur on different areas and might need to be altered, the sponsor staffing, how people actually can go in and do any of the modifications to the studies that need to be done. What is the support from the sponsor? Are they financially able to support this? Are they actually willing to work with the data requesters and the people that have participated in the data? How do we encourage them to support this effort? What is their investment or disinvestment in this area? Sometimes the projects might be completely closed, or there's no longer anybody who's working on that kind of area, whether that be that certain disease or certain organ in that company or university or whoever is sponsoring the study. So that might be another form of disinvestment that needs additional looking into. And what if there's a new sponsor involved? What does that look like? So we're going to get into all of these questions. And our first discussion will be led by Noah Seidman. He is a community and database manager for the BDSRA Foundation, and that's out of Ohio in the United States. And his talk is called Preparing for Retroactive Data Sharing in Batten's Disease, the BDSRA Foundation. Thank you so much, Noah.
Thank you. Let me see if I can steal the reins from you really quickly.
There you go.
Wonderful. All right, hopefully you folks are seeing that, and I am as well. Good morning, everybody, and thank you, Carrie and Krista, for having me, and thank you for being so supportive and patient as we put this together. I appreciate how quick you guys were able to pivot through all of the changes. So yeah, just to introduce myself a little bit, I am flying solo here, representing a coalition of folks that have come together to do both the clinical care side of Batten disease support, as well as the ongoing academic and research investigations. And also, I'm the guy in the room that doesn't have any letters after his name. So as per a couple of really great conversations before this, I'm going to try to bring a bit of the patient advocate perspective as well as the lived experience, because that is the other hat I wear. My life was in many ways shaped by a lysosomal storage disorder. My brother had MPS IIIB. And so I've been in this space for a really long while. Now, as the somewhat ironic title of the talk might indicate, we're really just on the cusp of getting started with a lot of this. So I'm going to talk through how this group of folks came together, what some of the guiding principles behind that are, some of the things that we're putting in place to really take advantage of the retrospective data sharing. And then at the very end, I'll break down and I'll sort of try to give my take on how, as a patient advocate, I want to interface with these communities and bring some of that into the conversation about data. Beg your pardon. So I'll start just by introducing our coalition. So we're calling them Centers of Excellence. I know that this is a really common way to bring a lot of disparate groups together. Ours are centered around care institutions, and so these are the folks that make them up, as well as the clinic director or primary investigator. So pretty diverse coalition, covers a good chunk of the United States, and I've been really, really blessed to be working with all of these folks who are not only actively pursuing research across the Batten disease community, but also represent the highest standards of care for our families. And I think that being able to have both of those perspectives in the room at the same time has really been a guiding principle in how we have some of these conversations, even about the very granular topic of data sharing. All right, so, and just to highlight that as this group came together and that process really started towards the end of 2023, a big pillar of that collaboration was to actively collaborate in research and data sharing. And I think that while there's a lot of other stuff going on here, a lot of other goals that this group has, one of the things that has dominated discussions in our twice monthly meetings now has been how we're going to effectively share data, how we're going to effectively advance research. And I think the reason for that is because all of us recognize that many of the aspects of rare disease are purely tactical. We're often looking at addressing what's in front of us today. And data and research really give us the opportunity to flex that strategic muscle, to be a little bit prophylactic, be a little bit planful, and really build some of these lasting institutions that are going to carry us forward into the future and make a much greater difference over the lifespan of our patient as we work together to advance research. And yeah, so let me pop over to the next and just give just a little more detail. So 9 centers of excellence, and one of the things that was really exciting to us as we were putting this group together is that at each of the centers there was an active ongoing study across all of the different subtypes of Batten disease, and there are quite a few. And in addition to that, a lot of these centers already had partnerships. So folks were already working together, already sharing and collecting data jointly. And that was one of the things that laid such good groundwork, is the folks that are participating in these conversations had entered this conversation with the mindset of sharing what we have is so incredibly important. I know that Kara emphasized that having data siloed just benefits almost nobody. And I think that one of the greatest benefits of this coalition has been that nobody is greedy or jealous of what they have. The goal is to share, and we're jointly tackling the challenges of sharing, which is one of the things that I want to chat a little bit about as we move forward. So there were a number of ongoing natural history studies in the Batten space. And one of the things that came from this partnership is an RDCRN grant application. Now, I believe the committee met, I think, last Tuesday, so hopefully we have scores incoming, but given the shakeups that have happened across our legislative and regulatory partners, I'm not really sure where that stands, and I think that that has both been a challenging point as a lot of us were sort of trying to have plans contingent on a successful application. But now we're having conversations around, okay, what does our data sharing look like in lieu of that? How can we build other things that will get us to the same endpoint, but might not have that explicit support that we had been hoping for? And so the first step in that process really was to do an audit of the different data sharing platforms that exist. And the reason I say platforms and not sort of jumping more directly into a study in an IRB and all of the things that come along with that is one thing that we recognized is there has a tendency to build atop the scaffolding of a previous venture in rare disease. And in many cases, that makes a lot of sense. You jump off from a higher point, you have all of the experience to build from. But I think the perspective taken here is that there's really something to be gained from being very explicit, being very planful, and laying a great foundation. If your goal is to cooperate into the future, then it makes a lot of sense to sort of start at the base level and build up from there. So that was sort of our first step, but recognizing that there is an immediate need in the community, the Centers of Excellence members also jumped in and said, hey, here is an exhaustive list of the ongoing studies. Here are the qualifying factors. We are going to bring our team, bring our phlebotomists, bring the folks that can collect some of this data, these samples, to the Batten Disease Family Conference and give families the easiest intro to joining some of those studies that we can imagine. And it was really, really successful. An extra hotel room and a lot of great coordination from some of our partners. And we got a lot of folks into different studies that they might not have had easy access to, either for geographic reasons or because they weren't aware of them. Let's pop out. So this one is a little bit more of a lighthearted take, but one of the early things that we did is someone said, hey, it would be really great if meeting minutes weren't passed around in an email attachment. Is there just somewhere that we could possibly put them all so that I can access them? And it turns out that when you have 20 some odd people across 9 institutions with different firewalls and different IT departments, it's really, really hard to find one place where everyone can access your documents. And so we had to turn to a survey to even figure out what the lesser evil was. And especially in this age of it being so very easy, it seems, to access a shared workspace, the fact that we couldn't hit a complete consensus and we were asking folks to take a longer route to get to something, even for the most popular, I think is really indicative of some of the obstacles that we face in looking at these problems of data sharing. And like I said, this was nothing more, nothing more private than meeting minutes or an agenda beforehand. And so now let's talk a little bit about sort of the meat of the decisions that have been made thus far. So looking forward, there is a lot of agreement on our pathway, and what we hope this looks like is a centralized IRB, either mediated by the organization that I represent, the BDSRA, or one of the constituent members. And we were incredibly fortunate to be able to adopt a structure that has been doing a ton of incredible work in Europe. I think probably everyone in this room has heard the case study of Brenura and BioMarin, and that was really driven by the natural history study conducted in Europe and following the structure, which is the Dem-Child structure. And the benefits of this is it's got a ton of great validated measures, a lot of good assessments for specifically for Batten disease, and is really, really well defined, widely used. And fortunately, our European partners were happy to just hand us this structure. And they put it very cleanly that there are two components. There's the static retrospective piece of the study and the prospective dynamic data that's being collected. And so made up in that retrospective data are things like demographics and genetic diagnosis. We've got medical history and a couple of measures. And so that gives us a really, really great wedge to begin to collect some of that retrospective data, to share it, and to take and adopt from pre-existing studies and say, hey, we have the structure, we have the formula. As long as we can go through this reconsenting process successfully, as long as we can modify those studies, we know exactly which buckets things are going to. I think data dictionaries are a wonderful tool, and because my preference is to frame things in terms of language, the more we have in that shared glossary, the more our vocabularies overlap, the easier it is for us to compare one-to-one and to successfully share not just now, but moving forward. So I wish I had something more concrete to say. I fear that the group is still very much in something of a nascent stage, but this is really where we are. And well, it doesn't necessarily feel like solid ground yet. I think that the shared values and the intention behind what is driving this forward is really clear to everyone. And so as we look for ways to enact that, I feel very confident in sort of the powerhouses that we have joined and our ability to find a great solution. And then before I close, I just want to talk a little bit about my own perspective. And I have a couple of brief things to talk about. And maybe this strays a little bit explicitly away from data, but hopefully I can bring it back. And the first of those points is, how do you engage with the bereaved members of the community? And I mention this because they are, in many of our cases, an enormous source of data. We know that the majority of our diseases affect children. And unfortunately, what that can mean is that there is a giant portion of the population that you have identified that are no longer actively caring. And so the obvious motivation to participate in studies is not quite there. And so how do we engage with those folks? And that's a question I ask a lot because I fall into that demographic as well. And I would say that, well, it is a really challenging thing. One of the benefits of sitting where I sit and where so many folks in our community sit is we do have that desire to be strategic. We are no longer fighting for the one, but our continued motivation, our continued resolve is to incorporate as much of the community for the good of as many people as possible. And so thinking really clearly about how we court that cohort of people, how we engage with them, and what they have to offer us, which by and large is an incredible amount of retrospective data, I think is a really great tool to gain a little bit more robustness, a little bit more of a baseline as we push to engage our actively caring families more and to really utilize that data, that dynamic data that's coming in. And the next question is that of patient ownership of data. I feel like this has been a really spiky subject to grapple with. Folks have a lot of really strong opinions of it. And I think just putting on my patient and caregiver hat is that the patient community is imagined to have far more concerns about data ownership than might be the case. I think that it is more a question of transparency that concerns us. I have found almost in every scenario when engaging a family for a study to do some sort of data sharing, to get involved in a patient registry or a natural history study, they are incredibly excited to be involved. But the way that we tackle this challenge of ownership and continue to be good stewards of that information is to be maximally transparent about what the goals are at the onset. And so the last piece I want to talk about, and this one just popped into my brain because I was helping a friend prepare for a talk, but the question was, when is the patient story data? And the two of us bounced back and forth for a while, and we landed on this idea that stories are data and the context. And so the question that I put to folks that have infinitely more experience and knowledge for conducting research and actually making use of data is, how and when is that context going to be important? And I will shamelessly steal her example that her son used to have these fits of laughter. And it was a great moment of joy in her life, because everyone loves when their sick child is just laughing. But one day she happened to post on a Facebook group for caregivers and say, hey, does anybody else's kid have these laughing fits where it seems like they just can't stop? And it turns out the answer was yes. And it turns out the answer that her son and many others were having seizures. And just that moment of a patient reported data, as well as a little bit of perspective, led to the diagnosis of a symptom and maybe even an opportunity to collect some more information and to be well used. And I say that because really the desire of patients, especially when they are reporting data, when they're reporting outcomes, is for that information to be well used. We are used to being cheerleaders. We are used to sharing our stories and sharing our data to be compelling, to be inspiring. But behind all of that, the pervading desire is that it be used for something actionable. And so thinking about that is one of the ways I think we can best engage with the patient community, how we can be advocates as well as scientists. And I think that's all I have. I will share the final slide with my contact information if anybody has any questions, and pass things off. Thank you, Kara.
Noah, we really appreciate you being willing to step out and to take over for some other people that may have spoken in your organization. But honestly, coming from your perspective, not only is it quite useful for others to hear, we have many just like you in the community that are finding ways to lift their own communities as well. So thank you so much for speaking today, Noah. Okay, so we are going to go to our next speaker, and 2 seconds to start here. And it is my pleasure to present Krista Kazaza, who is my co-lead on this project. She is the scientific director for pediatrics in the CPLD at C-Path. She's going to be talking about her work working with sponsors to encourage retroactive data sharing in lysosomal storage disorders, the CPLD experience. So thank you so much, Krista. I'm going to send this over to you and let you have your moment.
Thank you, Kara. First, apologies, both intra and interpersonal reason that I'm off video is, as some of you know, I suffer from vertigo, and today, of course, that reared its ugly head. And it just also happens to be a teacher work day, so I don't want my 6-year-old to rear her beautiful head. So I'm staying off video, but it's still going to hopefully be an informative aspect of specifically what we're doing at CPLD. So, hold on. I'm not seeing my slide. Give me 1 second. Kara, can you share my slides? Are they on there?
Just give me 1 second. I will bring them up.
Okay, sorry. I didn't know if it was my eyes or, I struggled a little bit this morning, so I'm sorry. Okay, thank you. Okay, so what we're going to talk about is kind of from the outside looking in side, kind of, and that is working with the sponsors to encourage retroactive data sharing, specifically for the lysosomal storage disease, in which we, as we mentioned previously. Go ahead, next slide. CPLD was founded in 2022 to, as basically a public-private partnership to really try to get a good grasp on the amount of data that is available and how best to utilize that data specifically for advancing drug development in lysosomal storage disease. We particularly focused on the unmet needs for regulatory grade solutions for neuronopathic lysosomal disease, and focused specifically on 9 neuronopathic lysosomal diseases that we identified as potentially being high capacity for data sharing. And so, that's a bi-directional aspect in that in order to develop the regulatory grade solutions, there has to be the level of evidence that would allow for regulatory grade pathways. So that, so it's kind of back and forth in terms of the amount of data that is available, but really what it's come down to is, even if the data is available, the opportunity for data sharing drives that that pathway. Next slide, please. So in general, the exploration of the regulatory pathway that we've specifically focused on was biomarker development. And as a lot of you know, some of you may not know, that there's no actual biomarkers for neuronopathic lysosomal storage diseases. And in order to address the highly warranted unmet need for neuronopathic LSD drug development, then we have to identify those biomarkers, which will serve as endpoints for therapeutic advancement. So we specifically have a one working group. We have 2 working groups. One is focused on clinical outcomes assessment, the other one on biomarkers, to look at the landscape, identify the gaps, and see what we need to do to ensure that the data available and the database within that specific LD is suitable to develop a comprehensive consensus delivered report so that we can move forward with the regulatory path. And that, it includes a letter of intent, a context of use, that type of thing in the drug development pathway for regulatory grade solutions. Next slide, please. And so again, we focus specifically on 9 lysosomal storage diseases that we, in the pre-consortium, the early pre-consortium stage, from the stakeholder input, and that includes industry, patient advocacy groups, and academic members, what their top prioritized diseases were, as well as the potential data available to be able to aggregate within our data analytics platform that Heidi's going to talk about in a few minutes. And so we started with the development of a biomarker landscape report with the 9 LDs. And this is kind of where we are in terms of the consideration and the tentative timeline. Within that working group I mentioned, we've sent 6 biomarker landscape reports. And then within that, we kind of summarized it in a table that we refer to as a dashboard to start to prioritize, based solely on the literature, what the biomarker candidates, the prioritized candidates are. Then with the working group, discuss gaps, the data available, how we can enrich the literature with the data available, and then do a gap analysis to determine where we are in terms of regulatory path for the biomarkers. Now, in full transparency, we were initially looking for 1 or 2 biomarkers that may be used, that would be maybe relevant in multiple diseases. That's obviously been more difficult than than we wanted it to be. Probably could have been anticipated, knowing that the work that's been done and the lack of biomarkers to date. But nevertheless, that was our initial intent. Next slide, please. So in terms of the development, we are working specifically within our platform, which again, Heidi's going to talk much more detail about in a few minutes. But we start with the data use agreements or data contribution agreements. And then with that, once we get the DCAs executed, then we curate and standardize the data. Again, I'm going to leave that to Heidi, who knows much more about the process. So we can start to analyze the data set basically in a metadata analysis, because we'll have the aggregated data and then be able to characterize the what we ultimately want in terms of the regulatory pathway, to characterize the diseases in terms of progression, based largely on natural history studies, but also in terms of trials, both those that are ongoing, those that are completed, those that are withdrawn or terminated, and what we see there. And so it's a multifaceted approach to characterize the biomarkers that are of particular relevance for the disease or diseases. Next slide, please. And just kind of what the data contribution agreement looks like. The start of the DCA process is an introductory call with the data contributor that we request, and we have a template, and then the contributor reviews the DCA and redlines it, and then there's internal and external reviews on both sides, and then the DCA is executed. Now, this seems like a three-step process, just back and forth, but obviously we know there's a lot of legal back and forth, and there's a compromise in terms of understanding, in terms of what is actually used. So the data contributor continues to own the data. We simply house the data in the DAP, and the data contributor has full oversight of who can access the data from, the minimal is the FDA and C-Path, and the maximal it would be full public transparency for the data. But the data contributor and the data contribution agreement makes that desire known, and it can be for a one-time data transfer or continuous if it's an ongoing study. Next slide, please. And so, some of the considerations obviously are the countries, that it's available. We have a GDPR compliant and a non-GDPR if it's just a national. The contributor name, who actually is the contributor and who owns the data, whether if it's an academic institution versus a advocacy group versus a industry. And so some of these things have to be considered, and whether it contains PHI or not, the data has to be completely de-identified, and the different types of data is in a drop down to be able to ensure that the considerations for sharing and transfer can be very transparent in terms of the transfer process. Next slide, please. What then occurs with our data managers is that the aggregated data set, again, the contributor determines if it's C-Path only, which is the FDA and C-Path. The consortium members, if they're only specific to the members of the consortium, qualified researchers, or the public, again, those are the different levels. And it still goes through a review with a request for the data to ensure that all of these things are being upheld. Next slide, please. So in terms of CPLD in specific, here is a snapshot of the current data outreach and sharing. And you see it's a multi-step process. It also is, it's not just we agree to share and then it's uploaded and then it becomes aggregated and it becomes available depending on the contributor. There's still some nuances that are somewhat limiting the from start to go point in terms of data availability. And I think I'll just circle back with Noah, and one of the most important aspects of that, once the data is transferred, is the data dictionary, as he mentioned. Oftentimes even within the same study, if it's different sites, they use different variable names in their data. So we have to have the data dictionary to ensure that when we harmonize and aggregate the data, that it aligns and that we're measuring the same variable and analyzing the same variable. Another aspect is, as Kara's mentioned from the beginning, the consent form. There's specific nuances within the consent form that need to be considered. For example, we are working with the MPC community in creating an international GUI ID so that we can aggregate the MPC data. However, with an international GUI ID that was created by the INDPR, the consent language specifically says that consent has to be explicitly stated that it would be shared internationally. And oftentimes the original consent form doesn't specifically mention international. And so there's specific words that actually can limit the use of the data. And in this case, the international GUI ID, while we still get the data, the process becomes much more difficult to harmonize and curate, because then we don't have the merging by international GUI ID, because if we have the GUI ID that was used nationally and they didn't specify, then we can't just merge them because of the specific consent language. Another and even more limiting aspect is in terms of academic data sharing. The data collected is usually collected on grant funding, and the transfer of the data oftentimes requires the de-identification or the creation of GUI ID that is not allocated in terms of budgeting. So then it becomes a manpower and financial limitation. So there's multiple reasons that this is slowed down, and why we're still struggling in some aspects, even with executed data contribution agreements, to get the data curated into the DAP. And so, we have a few different things, and there's multiple cases that we could discuss, but in general, there's a lot of challenges beyond just the desire of the contributor to share, that sometimes we can foresee, but oftentimes we can't foresee, and then we have to go back and we know that having to retrospectively go back to how to data share is somewhat more challenging than prospective. So starting with the end in mind is obviously what we try to do, but obviously that's not always possible. Next slide, please. And so, just in terms of what we're doing once we get that data, and, again, we're trying to identify these gaps and priorities, very similar kind of overview of what Noah talked about, in terms of it's just a different perspective. But we really want to be able to utilize this very valuable collected data from multiple stakeholders, multiple entities, to identify the gaps and priorities and determine specific endpoints so that we can, in fact, have impact on drug development and drug development tools. Now, these are very much anchored in natural history. This allows us for the use of external controls, in which we know that there's often not an opportunity or a good ethical pathway to use placebo-controlled. But it also allows us to support trial design, and it then in the long term allows us to link cross-sectional longitudinal landscaping, because again, when we develop these trials, the study design, the a priori knowledge continues to evolve, and what we know now we didn't know when the trials were designed, and that presents its own challenge just based on the level of scientific insight that we had at the time. So it allows us to be more flexible and perhaps extrapolate data, perhaps utilize, again, external controls to be able to aggregate similar diseases, even if the progression is different, but the biomarker or the response to treatment for some of these basket trials or other things like that. It allows us to hopefully make decisions about endpoints, which is severely lacking in lysosomal diseases and has a huge impact on therapeutic options. Next slide, please. And that is the end of my slides, but I think that it segues nicely into my colleague, the scientific director for the RDCA DAP, Heidi, who's going to actually give kind of an overview of this mysterious platform that I've talked about, because she knows it better than anyone, and I will turn it over to her.
Go ahead, Heidi.
My apologies, I had a hard time unmuting. Thank you, Kara and Krista, for inviting me. I'll get back to where we started. I'm going to go over high-level overview of the RDCA DAP platform, which was highlighted a bit by Krista, but we will dig in a bit more on the governance aspect of things. C-Path is a pre-competitive neutral convener, and the idea is to develop these public-private partnerships, which has been done well with the CPLD community, with the idea to aggregate data, retrospective data that is de-identified, patient-level data, to hopefully come up with the best science, the broadest experience, and active consensus building to both share in the risk and the costs to overcome bottlenecks in drug and therapy development. And in doing so, we hope to have industry and regulators, patients, academia, and the government at the table. And this helps to enable iterative regulatory participation in developing new methods to assess both safety and efficacy of medical products. And the end goal is official regulatory endorsement of novel methods and development of drug development tools. So we have discussions, as you all have had, with the patient communities of their unmet needs, but also with the industry partners of what their unmet needs are in their therapy development pipelines. And we leverage the core competencies within C-Path, which includes these solution-specific consortia that have been developed within the biomarker space, modeling and analytics, data management and standards, and clinical outcome assessments and regulatory science. And utilizing these competencies, the goal is to develop future tools, which Krista spoke to. And just quickly, I'd like to highlight some of the wins that we've had using this model. And that is authorization of some of these tools by the regulators. In Duchenne muscular dystrophy, we had an EMA letter of support for a clinical trial simulator. In polycystic kidney disease, we had EMA and FDA qualification of a total kidney volume imaging biomarker, and in transplant therapeutics, the EMA qualification opinion for the iBox scoring system. So there have been several qualification decisions, letters of support, fit for purpose endorsements with the FDA, but also qualifications that have gone into the European regulators and the Japanese regulators.
Hey, could you please go into presenter mode? We're not seeing your slides completely. Thank you.
I think I'm in. I can start over.
Oh yeah, try one more time. We just weren't seeing your slides advancing.
Can you
There
see
you
them
go.
now?
Yes, perfect. Thank you so much.
Okay, so I apologize about that. So RDCA DAP, as I had said before, is a independent, neutral convener collaboration with analytics that promotes the sharing of critical data across rare diseases in order to accelerate and understand disease progression and optimize clinical trial design. So ideally, the data that comes in is curated and standardized using fair data practices to encourage further standardization and allow access to that patient level data as part of the integration of data sets. But there's analytics built into the workspaces of the platform that allow users of those workspaces deeper understanding of the data, and the scientists that work for C-Path the ability to build model-informed drug development tools and solutions and further understanding of those rare diseases to hopefully develop solutions that overcome current bottlenecks that are holding up therapy development. These are the current large consortiums that are housed within RDCA DAP, and you can see CPLD is one of them, but there's several others, and there's smaller scale task forces that we're also launching. But the key thing that I wanted to talk about was the data contribution agreement. Now, this is the legal document that dictates how a data set is shared. And Chris has spoke a bit about how we reach out to a contributor and the DCA is shared back and forth and the contributor is allowed to redline it. But it tells us that the data was collected ethically and that there is patient consent that allows for the sharing of the data, that the custodian is able to share the data, and that it will be de-identified, because that is required for sharing on RDCA DAP. It encouraged data sharing in a flexible way, in the sense that the contributor retains ownership and they're able to share in a number of different ways that suits them best based on their incentives. So they can share with the consortium and with the scientists for the development of tools. They can share openly. Oftentimes foundations want as many scientists to find their data and utilize it for future research, where a company may be very interested in being part of a consortium and having the feedback on their data collection and having the data used with other data sources to inform data tools, but they may not want it open to everyone to come and put in a data use request. So we make it as flexible as possible to get as many data sharing contributors incentivized to share. They also are able to share, I should go back, associated documents, so an IRB or a protocol, so that then people who are academics sometimes want people who are looking at their data to fully understand how that data was collected. And a protocol makes them feel more at ease sharing their data if that is attached to the data set. Now, the data access, key takeaways I'd want you to know is, as I said before, the custodian will maintain ownership, and the recipient agrees that it will restrict its use of that data set to the analysis that they put in their research plan when they request data use. The standards and safeguards are enforced when we grant usage of data. A contributor can request that it goes through our data usage committee, but they can request that they also vet those requests that come in. So whether it be a foundation or a company or an academic, they could be the final say of whether that data goes to the requester or not. And publication. C-Path may share the research plan with the data custodian if that's how the data custodian requests it be shared when the data request comes in. And the requester, and I've covered this, recipients shall submit, for example, within 30 days that citations and acknowledgements are appropriate. And there's no scientific review, but C-Path may share publication to the custodian for comments. RDCA strongly encourages public dissemination of all results of the analysis that's been done on any data. And then just to reiterate that this path of data coming in, it being analyzed and feedback to the custodians, the standardization, we like to define the core elements, improve collection practices, the data aggregation enhances information and analytical value. We try to avoid duplication of research efforts, improve knowledge for disease progression, patient variability, enrich clinical trial methods, stratification. There's a possibility of endpoint measures and biomarkers being identified utilizing this type of data. And then, of course, development of drug development tools. That's it for me. I can stop sharing. This is just a contact information if anyone is interested in sharing data.
Thank you so much, Heidi. We really appreciate you sharing information about this important resource that many of us use. So thank you so much. Now we are going to turn to our next presenter. One moment. Okay, and so here we have Janine Jarnes, who is a PharmD MSc in BCOP, BCPS, which I'm sure you have a lot more to say about that, at the University of Minnesota Department of Pediatrics in Minneapolis, Minnesota. And she is going to be talking to us about the natural history study of gangliosidosis. Thank you so much for presenting for us today. Thank you, Janine.
Okay, I was having a little trouble unmuting, so let me just make sure the slides forward. Is that, oops, okay. So okay, there, I can forward the slide that way. Okay, so I will be talking about a study for which I am the principal investigator, and this is the natural history study of the gangliosidosis. And I work at the University of Minnesota. And this study has been open since 2009. It was part of the Lysosomal Disease Network collection of studies that started at that time. at 12 different centers of excellence for different lysosomal diseases. My study, some of the studies were fully funded by NIH within this consortium. My study for the gangliosidosis was not fully funded. So I received some partial funding that brought in funds for travel expenses and lodging. But all the clinical evaluations I did, none of them were funded. And so we were flying patients in and we were trying to get their insurance to pay for different clinical evaluations as part of clinical care. And it's a great study, and we also will accept patients remotely if they can't travel. But as you can see, since we're depending on insurance coverage and limitations on travel, we get sometimes different data sets for different patients when you compare them. So it did create a bit of a challenge for me initially on how do I publish on this data. But we have about 60 patients enrolled at this time. That's a nice number. We do have an international group of subjects. Most of them are from the United States, but some are from outside the United States. And it collects prospective data, but also retrospective data. The study covers both GM1 and GM2 gangliosidosis, and it covers all phenotypes: infantile, late infantile, juvenile, and late onset. And here's 9 of our publications that came out of this study so far. I would like to mention with what Noah said about patient advocacy groups, the second publication there was actually a patient advocacy group publication through Cure GM1. But I contributed to that publication based on my natural history study. And I think it's important. I just want to put in a pitch for patient advocacy groups to start working on getting peer-reviewed publications out, because I think it empowers them and brings more visibility to them. But Noah also asked about bereaved patients and how do we get data from families who have been bereaved, or bereaved families, I should say. This particular study, the second one, Bingham et al., we were mostly interviewing and collecting data from parents who had a child who had passed away. So it does represent work done with bereaving parents. And there's been a few other studies over the many decades that have done that in the gangliosidosis field, maybe 1 or 2 at the most. But as Noah said, with these small populations, oftentimes we do have to approach parents who have lost a child or family members who have lost a family member, see if they're even willing to talk about it. Are they okay with us collecting some data? And it also creates a memory. Sometimes things aren't remembered quite as clearly, even 1 or 2 years after the patient has passed away. So that has to be taken into account, but it's very valuable data. So we started a data collaborative effort, and I'm an advisor for Cure GM1. And they focus on GM1 gangliosidosis. And they joined up with C-Path, trying to get different stakeholders who have natural history data to contribute data to a collective pool so that we can get better data that's maybe more robust with more patients, and kind of see what everybody's doing. As we've been talking here, as you guys all understand, it's very important if we can pool our data, especially in rare diseases. And I believe it took a couple of years to get this collaborative agreement signed. It was a long process. It involved a contract between the regents of the University of Minnesota and C-Path. And as you've heard from 2 speakers on C-Path, it's a nonprofit organization. They house the data. They use a data and analytics platform, the RDCA DAP, and potential stakeholders, as you've already heard, or as you've already gathered, might be academia, patient advocacy groups, industry, regulatory authorities, and government. And in our particular case with GM1 gangliosidosis, the academia is a part of this collaborative group that is working with C-Path. And right now we have 1 patient advocacy group, Cure GM1. We also have industry stakeholders that have been actively talked to during this whole process. I believe Krista maybe mentioned, I wrote them down because it looked like for industry, we might have Invitae. I did not really see any pharmaceutical companies on there, Krista, yet, but they have been involved in many of these discussions with C-Path. We'll see how that plays out. I know there's Passage Bio, there's several other pharmaceutical companies that would be considered, you know, very much involved in this disease. Dr. Roberto Giuliani from Brazil is also a contributor of natural history data. But he is, again, in the academia realm. So regulatory authorities such as the FDA gets involved in this. There might be government funding towards C-Path that helps with different projects, as you've already heard some discussion of. So important definitions, and I am not an expert with these definitions, so I actually made this table, this page to help myself, but it's really important to understand the data when they say data on this agreement that I made with C-Path. So I had to sign the agreement with C-Path. I had to work with the regents of the University of Minnesota and their legal team to get this all worked out. But data is what I'm contributing. The research is any research used for the advancement of the goals of the collaboration. So that's kind of a broad scope. The research results are not my research results. They're what come out of this collaboration of all the data coming together. And you've heard Krista and Heidi talking a little bit about data access requests and talking about how that works. The metadata, to my understanding, and Krista and Heidi might want to correct this, but it's really an initial kind of something that the C-Path might, if someone comes to request data, or a data requester, C-Path, they might say, what kind of data do you have on GM1 gangliosidosis? And so C-Path might have a metadata, which is sort of a way to say, this is the type of data we have. It maybe we're looking at the Bayley's infant scale of toddler, or scale of infant toddler development, or we're looking at the Vineland, or we're looking at imaging, MRI and MRS results. That's the type of data we have. So without giving out individual data. And then the summary data is also data that is a product of what C-Path is doing, summarizing different sets of results. And the approved users, this was basically from the C-Path website, or actually from the contract and the C-Path website. It may be the C-Path staff and persons that C-Path is using to help with their whole project. But going back, even I'm thinking about what type of data am I going to contribute to this? Okay, I haven't yet done it because I'm organizing my data. But what's really important to me when we're doing data transfers into a collection pool of data from different institutions and different groups, whether it be patient advocacy groups, et cetera, what types of questions should we be trying to answer with this data? And we may think that's obvious, you know, what happens in the natural history, et cetera. My study, the natural history of the gangliosidoses, specifically was given a mission by NIH. Your study is for clinical trial readiness. So you're doing this study in order to find data that will help eventually lead to clinical trials. In other words, the data I have in my interpretation of this mission I was given, the data needs to provide meaningful clinical outcomes that pharma can use to do clinical trials for therapies. And one of the things that I discovered in my study, I just started kind of randomly. I had a protocol. I was trying to get everything collected in terms of the data that I promised to collect on my protocol and trying to document that. But what I noticed as I interviewed these different parents coming in with their little child from all over the United States and some from Europe, and we had a patient from China, and we have patients coming from some other countries too, England. By the time they got to the University of Minnesota and traveled all that way, they were motivated to come because they just found out that their child has a disease that the child will die from. It's uniformly fatal. My child is going to die. This is a catastrophic disease. So they already know that. when they come to us because they've already looked. They've already had some pre-communication. They've worked with their local physicians. There is no treatment. So my child's going to die. So why am I going to Minnesota if I know my child's going to die? Why am I coming all this way and carrying a sick child with me? That there's risks in travel. So what was curious to me, which I wasn't even looking for, is that I noticed every parent said, the reason I'm here is, number one, are you working on any treatments? Do you know if there's any treatments coming that might be available? Will it be available in time for my child? Lastly, I'll do anything for my child, but I know the child's probably going to die. So can you tell me what's going to come next? My child never learned to crawl. They just were diagnosed with seizures. Everything that happens, every time something new happens to them, it's very shocking. And my local doctor's never seen a patient with this disease. They can't tell me anything about it. They just say, try to keep them comfortable. What should I be preparing for next? Do you know? Well, when I first saw these patients the first few years, I tried my best, but I went through the literature and I thought, well, all these things can happen. I don't really know what's going to happen next, but here's what can happen. So an important thing that I discovered doing this study, and I think this applies to C-Path, and that's why I'm talking about all this. It applies to how we pool this data. It's not just important what happens in this rare disease. None of us know what all happens, right? We're trying to figure that out. So when does it happen? And shockingly, I really didn't see much of that in the peer-reviewed literature. And I also did not see anything in the peer-reviewed literature saying, this usually happens first, and then oftentimes this follows, and then this follows. Why is that so important? There's two reasons. Just out of compassion for these parents and their life, they need to know what to plan for. They were asking me to tell them, what do I plan for next? Also, for clinical trial design in these rare diseases, if we're going to have meaningful outcomes, we have to have an idea. Is there any pattern we're seeing here where we could at least say, a lot of this stuff is heterogeneous. We don't know when it's going to happen, but these things here, this happens and then it seems this happens. Or this seems to be the first thing that happens that brings someone to look for a diagnosis in the infantile phenotype or in the juvenile phenotype. These are important things to discover and to document for pharmaceutical companies developing therapies, because then they have a timeline in which they can say, we know all these things happen within this first and second year, or here or there within these set of years of this child. Or we can even say this usually happens first, followed by this. And this might create some initial, if we can't find really great biomarkers to use as surrogates for the time being, something to measure to see, is the drug therapy working? So that is something I want to share, and I want this to happen with C-Path, not just say what happens with the disease, but is there a sequence of event, any type of pattern we're picking up on this? It's incredibly important for bringing therapies to market. And I coined this the timeline of clinical change. This is just a chart from a study on the infantile patients, both GM1 and GM2, where I was starting to see patterns. So a few years into my study, I was actually able to tell the parents, this is going to happen, this is going to happen, this is going to happen, this is going to happen. These things are going to probably happen in this order. The other ones, I don't know what order, but they're going to happen between the age of 1 and 2, or they're going to happen most likely between the age of 2 and 3. That was incredibly valuable to the parents, but it's also good for clinical trial design. Here's even just simple standard pediatric developmental milestones. We discovered those can be used as outcome measures in the infantile phenotype. So, and also defining infantile and distinguishing it from late infantile. So that is important, and I really hope this work with C-Path continues looking at those things and getting more data to make it more robust. Ownership of the data, as you already heard from Heidi and Krista, the contributor retains ownership of the data. That was something that I was very important to me. And I may, if upon my request, I can tell C-Path, I want to know anyone who requests this data, and I want to be part of the decision on whether or not that data is given to a requester. The approved user of the data retains the ownership of the research results. So I am giving up a little bit of control here, right? And I've had other colleagues that I work with say, don't give up that control. That's your data. You worked so hard to get that. Don't give up that control. The reason I chose to give up the control is because this is a uniformly lethal disease. There are no effective treatments right now. So in my view on this whole thing, and in my lifetime, and I'm a senior person now, if anything's going to happen, I'm going to have to give up some control and contribute. And likewise with the other data contributors, we're going to have to give up a little bit of our control of the data, but we can write this agreement with C-Path in such a way that here's the stipulations I have. I want to approve any publication that's using my data. I want to see what you're doing with the data. I want to see how you're analyzing it. I want to give you advice, and you missed this or that, or let's make sure we include this or that. And based on my discussions with C-Path in the initial part of this, they're very willing to do that. So I said, well, let's move forward, because me just holding this data here, it's helping. Pharma companies are calling me. I'm helping them design outcome measures. So that's great, but it's going to move much faster possibly if we do a data sharing. So what kind of consent was required at the University of Minnesota? I was thinking I might have to reconsent patients, and back to Noah's discussion of what about bereaved families, I would think it'd be very difficult for me to contact a bereaved family even a few months out, but 4 or 5 years out after they lost their loved one, and say, do you mind going through a consenting process again? We're going to share this data. Let's go through the whole consenting process. I'm not sure how successful that would be. That might be quite challenging. And so luckily I was pleased. The University of Minnesota said, you don't have to reconsent these patients, Janine, because your data is completely de-identified. You're contributing de-identified data. And right now the data I'm contributing, I've already published on it. So basically I'm reorganizing what's already out there in the public into a format that can be maybe more useful for C-Path with the project they're doing. Will I need to reconsent patients in the future? Yes, possibly I will. Okay. I'm going to approach that depending on what kind of data we move into, if it gets more involved, I might have to raise my hand and say, wait a minute, we might need a reconsent. C-Path sends all data requests to the governance committee, and I'm really excited about this governance committee. I will be the person representing my study on the governance committee. And that gives me some feeling of control over this in terms of how the data is interpreted, who accesses it, how we publish it. The governance committee, according to my understanding, is there's a representative from each data contributor. Those are some of the members, and a representative from each stakeholder in this collaboration, whether it's pharma, whether it's the patient advocacy group and C-Path. And all data requests must be approved by the governance committee. So again, that gives me, at least I have a say in what happens. And Cure GM1 may request additional information from a requester, and they may also decline the request. So there's a certain amount of control retained by the different parties contributing data. And again, that is reassuring to me. My main concern is I don't want the data to be misrepresented. I don't want it to be misinterpreted or misrepresented. What about cost? When we started this talk with Cure GM1 and C-Path hired, they had an attorney working on it. Cure GM1 wanted a cost to be involved. They're a nonprofit organization. They're struggling to support their mission and cause and goals. And they wanted pharmaceutical companies who may have a little more resources available to contribute to maybe to Cure GM1. That was my understanding. Or maybe it was C-Path, maybe Krista and Heidi can clarify, if data was accessed. But at this time, there is currently no cost to accessing the data. So that's where it sits right now. There was discussion back and forth, and I know that the pharma companies were not happy with the proposal in this situation, that they would have to pay to get the data. But as things change, and we'll see how this all goes. You know, maybe there'll be situations where it really makes sense for them for somebody to be putting money into it to help pay for the costs of this. Publication plans, again, I was talking about the governance committee. They'll have a publication committee. And I think Krista and Heidi talked a little bit about this, you know, that the contributors can approve these publications. Now, maybe not all contributors care, but I made it really clear in my agreement, I want to be able to approve any publication. I also need to be a co-author. Or if I decide not to be a co-author, I need to be able to make that decision. So what are the staffing needs and the resource needs at the University of Minnesota to do such a collaboration? We currently have no funding to do this. We're doing this, you know, because it's good work. And I am the principal investigator. I will be the one resorting the data and giving it to C-Path. Our grant and contract officer from the Sponsored Projects Administration was involved in the initial setup of this. I will also be working with that person as I go to contribute the data. And the Sponsored Projects Administration in general was involved at different legal and other administrative levels. When I first started, I'm almost done. When I first started talking to the grants office, because I'd never done this before, we're like, what type of agreement is this? And it's not a materials transfer agreement. There's no tangible material being transferred. And we decided it was a data use agreement. They decided that. The research collaborative agreement, that sounds like that would be it. But my Sponsored Projects Administration said, those are usually involving intellectual property. There is no intellectual property at this time in this. So we went with a data use transfer agreement. And I had to notify them that I wanted to do this. I had to send them my protocol and information from Cure GM1, and C-Path sent them the initial agreement to look at. I had to submit information into something called Minnesota GEMS or MinGEMS. I think every institution has something like this. It's an electronic system that you put in information on grants or different projects you're working on, and then it goes to the equivalent of the Sponsored Projects Administration. Eventually, I signed the agreement. So is this a sustainable effort? I don't know. But if we don't do something, nothing's going to happen. So we're all moving forward. Everyone here on this call, I think, is moving forward in their own realms of what they're doing with rare disease research and data contributions to a housing, you know, someplace that can house the data and help them disseminate it in a meaningful way. So I don't know what the long-term sustainability is for any of these stakeholders I've listed here, but we're forging ahead, and we're hoping for a very successful project here. So that's the end of my talk, and I will stop sharing.
Thank you so much, Janine. We really appreciate that deep dive into gangliosidosis and the really important work that you're doing here with Cure GM1. And just as a spoil alert, we actually will hear from Cure GM1 and their approach to the collaborative. So you'll learn a lot more about how that started, some of the challenges that they focused on, which Janine was able to point out here. That's actually my experience as well as working with Cure GM1. So Janine and I have worked for a while together on this project, and it's exciting to see that not only data is being deposited, that we have a next step for this. So thank you so much for that presentation. So I'm going to just turn us all now to our, here we go. Oops, sorry about that. I'm going to turn us now just to our Q&A. So if I could get everybody to come back on, and I'll just give here a little, so we can see here, these are the groups that are going to be talking again. We welcome back Noah, Heidi, Janine, Krista, and I am here as well. Krista, did you have something you wanted to say? Your hand is up on your...
Yeah, I just wanted to clarify in terms of cost to and from C-Path. There's no cost to and from C-Path, and when the DAP was created, it is FDA funded, and there's no cost. I can't speak as GM1, and I don't wish to, and Christine later can talk or not, but there's no cost. There is, in fact, in our model, the public partnership model, the consortium members, pharma does pay a membership fee to be a part of the consortium, but there's no cost within the DAP itself. I just wanted to clarify.
Oh, I can clarify from CareGM1's perspective, there's currently no cost for a researcher to request accessing data, and one idea that CareGM1 bounced around with the companies was an initial cost to support the effort overall, but another idea was thrown around, which was to not really thrown around, really debated at length, was a back-ended approach where data, if used successfully in registration, and that there would be a formula based on the success of the use of the data, that as like a kind of a success fee, because a lot of people have fears in general, the companies, that the data will not ultimately be useful for them and they'll be paying for something that isn't useful. So that's where that idea came from. But because unfortunately the biotech companies at the time felt this would kind of take too long to come together for them, we do not have any biotech sponsors, so there's no money changing hands right now, as Janine said.
Thank you very much, Christine, for jumping in on that. I am actually going to stop this share so we can be on camera as people ask questions. We have a couple of questions from the Q&A section. I don't know if it spoke to anyone specifically, but I can just start. I'm actually going to start at the bottom here, but we'll get back to you, Teresa. I'm sorry, no, that was just a comment from Dean saying, thank you, Janine, for being so transparent about helping patients and families and along quality of life alongside solid research. And I second that as well. Thank you, Janine. And yes, the comments on relaxing control of data are appreciated too, which is actually really interesting. And I think I do want to start there. So a couple of people, specifically Noah, you had spoke to the idea that a patient's idea of privacy and control are actually more the balance that you're looking for versus just and transparency. Can you speak a little bit more to that? Because I think that is actually kind of enlightening for some people. I think especially when we had our presentation at the beginning, we spoke very heavily on ethics and privacy. And I think sometimes from a rare side, it feels that privacy is so important that it keeps everything else from moving forward. And in rare disease research, it's a really different calculation. Perhaps you could speak to that, Noah, and everybody else can join in as necessary. Go ahead.
Yeah, I would say that from the patient and from the caregiver's perspective, a lot of the sort of abstraction that lies between the data that I'm giving and the endpoints for that data are what become the primary cause for concern. And so I think that rather than restricting access, being significantly more constrained in how you're able to use things, if you position that effort to make the patient community well informed and well consented, I think that you will find they're more willing to agree to a greater variety of things. Because I guess from the patient perspective, there's no interest in having ownership of the mechanism or the inputs. They really want the ownership and the participation in the outcome, in the potential treatment or cure. And so if you take that mindset as you're talking to patients, sometimes it could be a little bit easier to grease the wheels and to reach an agreement that allows their data to be maximally useful without sort of impinging on that sense of ownership.
Yes, and I think actually also what Dean was referring to is not just the patient, but I wanted to bring it back to that, but also the PI's control. And Janine, I think you can really speak to that, because I think that was really enlightening as well to hear that yes, there's some amount of control, because of course publishing is, our method of transaction at this moment, until we come up with another system that better tracks our output. And to see that you found a balance there was really enlightening. Ginny, do you want to expand on that a little bit?
Yes, thank you. When Cure GM1 and Kathleen Kirby and their group brought this up, they tried to get all the stakeholders on these very large conference calls, again, about 2 years ago, and everyone agreed it would be best if our data was all pooled, right? But again, in academics, there's different reasons for wanting to control the data, or the data, however you want to say it. And I was actually told by some people I asked, I said, this is a great project. Here's what I'm talking about Cure GM1 about. Do you think there's going to be a problem with sharing data? And I was told by several leaders in the field, don't share your data. Do not share your data. That's your data. It could be misused. You own that. You're going to lose complete control of it. That type of conversation. And as an academician, we get evaluated every year for in academics on how many peer-reviewed publications we have out, how many peer-reviewed publications we're a first or last author on, et cetera, what kind of journal it goes into, how highly rated is the journal. It's a big part of sustaining your gainful employment as an academician. But the way I looked at this was there's got to be a way to do this because it's so important. So you just have to put, in my view, I was like, okay, I'm going to move forward with this. And if I get any legal pushback, I'll have to deal with that with the University of Minnesota when it happens. But I'm not going to sit back and just list all the reasons why it won't work. I'm going to move forward and consider this uncharted territory for me, as it might be for others, explore it and just see how far we can go. And it did take a couple of years. And I think Kathleen Kirby was like, is she ever going to get this set up? And she was very gracious to me and very helpful. But we did get the agreement in place. And now I need to get the data to them, which will be this summer. I've had some delays. I'm doing some compassionate use IND projects that have delayed things. But I guess to summarize it all, Kara, I'm going on and on here. We have to think about the kids and how are we going to get cures to them? And will sharing this data make treatments become available more quickly? Will sharing the data bring good clinical trials into action more quickly? If there's a reasonable confidence that that's going to happen, then my feeling is I have to share that data. I have to figure out a way to do it. And right now there might be certain restrictions, but I have to do what I can. So I'm sorry if I went on too long there. I was trying to explain my mental process on all this.
Not only is this extremely important, these are discussions, and I'm going to get to Krista in 2 seconds, that I know Krista's having constantly. And that makes it a real challenge because the way that we have in the past and currently still do evaluate success is mostly on the publications that we put forward, and the next grants we get are based on the publications that we put forward. This cycle does not account for the outcomes in actual treatment, which would actually make the most sense. And we would hope to, in this era of reform and streamlining and efficiencies, it would be great to see PIs recognized for the advancement of the work that they do and the treatment that does occur. And the more credit that we could get to that back to the grant system and the tenure system would really help all of us. And I think that the more of us that speak up like that and the more communities that are touched in that way that can give that feedback, I think the best for everyone involved. So thank you very much for bringing that forward. Krista, did you want to add to that? I know you have so many rich experiences.
Well, I mean, I just wanted to highlight the work of Cure GM1 and Janine, because this is kind of how we envision the CPLD consortium. And indeed, there is some aspects of competitive and professional development, but we're working through those in terms of with our governance and publication, and really the PAGs driving, the patient advocacy groups driving this, because it is, as Noah said, it's their data, their story, and the outcomes are so important. We're moving towards slowly, industry trying to come out of those competitive barriers. Publications, as an incentive is a lot easier than the financial gains in which the industry may or may not leverage in sharing their data. So we're moving slowly but surely in terms of that. But I just wanted to highlight that in principle, the relationship between GM1 academics like Janine, and similarly across other NIH entities that we have really strong relationships with, that is starting to push the needle towards at least our initial mission. And I think as these walls come down in academia, and driven largely by patient advocates like Noah and Christine, that we'll start to actually get some of the data that can inform trial design, which is what Janine's ultimate goal is. And so it's a win-win-win for everybody.
Thank you, Krista. That it is really, it's wonderful when we all start working together towards this shared goal, and we really do anticipate great outcomes. I would like to actually touch on something that both Janine and Krista just mentioned, and it has come up a couple of times, and we're not going to be able to solve this problem, just to end that before it starts, but engaging with industry is a bit of a challenge. And we have found, even trying to engage for this presentation series, it's a little difficult to get people to talk about what they're doing internally. We do know that we have examples of great people at different institutions that have been able to go into their industry-sponsored and natural history studies or registries and have been able to get that information out and shared. It's been mostly times, at least I've seen, that the programs have been closed. So there is no longer an interest in that work, and it makes it a little bit easier. But of course, we are always finding and looking for ways to encourage these industry partners to share. I do know, because this had come up as well, talking about having industry fund some of this work. And the reason that I think really that comes to it is because they are the ones that benefit from the clinical trials when things do occur. And that does make sense in our current infrastructure. Now, if we want to look at this in a slightly different way and reimagine these infrastructures, then perhaps we have a space that doesn't include that. But at this point, both as Janine had mentioned, funding for a lot of the work that she's doing, she has to find other places to do that. And it's not coming either. That kind of work needs to come from the government sponsors that we have, or perhaps the nonprofit sponsors that we have around, though we know that those groups are included, are a lot more limited. But to have a focus on how do we get the data out, not just how do we create the data, not how do we engage the data, but how do we fund the next steps of getting the data out, is something I would personally like to see a bit more of a focus on, and we definitely will bring this up in our resulting publication. But does anybody want to speak to their experience with industry at all? Not trying to cause any challenges, but if there's any insight and maybe something that we could help that would inspire others to share within industry. Does anybody have any thoughts about that? Not trying to put anybody on the spot here, but if there's something to share. Okay. Yeah, that's
no,
okay.
I do, Kara.
Yeah. Oh, Christine, thanks. That's great. This is Christine, by the way, Wagner from Cure GM1. She spoke up earlier, but just in case people aren't aware of who she is.
Yeah, so I'm Christine Wagner. And the issue I found with this, and it's a long process, but it is that people want the proof before, and as you're Well, so we were in the process of assembling the data set, and the companies want everything finished in front. So the investment as you are building this is very hard to get. It may be something that you maybe just have to do yourself. So I totally believe that the companies will come, but they kind of want the finished product, which is a hard thing as you're trying to put this very complex project together.
Thank you, Christine. Noah, did you want to add something?
I would just say that, and they're also looking for a guarantee that this is clinical trial grade, fully approved and recognized data, and that can be a high bar to clear, especially as you are marshaling the resources to get started.
Very true. Very true. We do occasionally, like I said, have industry sponsors that create clinical trial ready material. I am aware of a couple that I am intimately involved with. So I think it's a little different when it comes from that end. And then, like Janine was mentioning, there's some industry sponsors that we had talked about in the past. And sharing that data, I think previously had been done in a licensing way. So having this, a different approach where it's more of an open data sharing platform does kind of change the dynamic a little bit too. Again, we're kind of rewriting the scripts on a lot of these about ways that we encourage to share and to share. But it would be wonderful if, once this does come along, that people are sharing widely. And yes, licensing can still occur. We're not saying you can't license your data and that you can't have these other levels of interaction with data once it is shared, as Janine had pointed out, which is really a great example of how you don't lose control once you share the data. You actually just give it another opportunity to move forward and to impact more people, especially in our rare community. We know how important that is. Okay, great. So does anybody have anything more to share on that?
Just
Go ahead.
to Noah's point, as you speak to industry partners, I think the criticism they have is the quality and quantity of data available is not up to the standards that they require for the development of their product and what's going to help them moving forward, right? Whether it be natural history or registry. And so developing deliverables that will entice them as in utilizing these models and how we can bring them together is sort of the key, and thinking about our clinical partners that have spoken, because what you'll see is those same industry partners either fund their own natural histories and spend the money themselves because it'll be much more cost effective for them to not have to pay for it themselves, or help fund the ongoing natural histories by the academics because they need it one way or the other. And so it's a matter of selling and developing what fits their unmet needs, right? And what is available and how can we make that data more regulatory grade, as Noah said.
Go ahead. Oh,
Yeah, I just wanted to point out, I can't do it justice, but there was a, excuse me, there was a talk in our International Neonatal Consortium on Friday by an FDA colleague. And, what I want to bring about is she spoke specifically on the extrapolation of data in pediatric studies. And so many of her examples or in her case studies rang true in terms of what we're doing. And why I bring that up now is because some of the approvals, 3 of the approvals that she used as case studies, oftentimes the data that was collected wasn't intended to be used for what it was used for, but it was it came about that it was actually could be extrapolated and aggregated across different staging within pediatric disease. For example, congenital heart disease approval. And so again, I very much validate both Noah and Heidi's express concerns from the industry. But oftentimes we don't know what we have until we know what we have. And so that, that also requires some flexibility. And so I do encourage, I think we have that available. We'll have that available on our website. But it kind of changes the paradigm in the way you think about how data can be used when you think about extrapolation and, just ways in which we can take novel approaches. Again, with C-Path's facilitation of the approval for Friedreich's ataxia with the propensity scoring. So, the more data that we share, even in the purest forms of how it's collected, the more opportunities that we have. And I think that we need to help encourage industry to view things more in a creative manner rather than just what precedent is. So because if we only look at how precedent is in terms of regulatory pathway, then no drugs will ever get approved. So I mean, it's just a different perspective.
Noah, did you want to add something?
Yeah, I was going to echo a lot of what Heidi had said and say maybe, to bring a whole nother framework into this, we need to think of this a little bit more like a game theory problem and emphasize that this isn't a single instance of data sharing, but this is a collaborative partnership that's going to go out into the future, right? The horizon on a natural history study for a lot of these rare diseases may feel long to someone who's actively caring, but on the scope of a researcher or an industry partner, they're thinking decades in advance, and that's how we need to frame this.
Thank you. That's great. I really do appreciate and really am looking to see what these new creative ways of thinking. And that's what it's so great when we get people from very different backgrounds to have this kind of conversation together, because we hear about different experiences and learn a lot more about where we can go next. So that's great. Thank you so much for that discussion. We have a couple of questions about registries and natural history studies, as one would expect. So there's one from Teresa that says, is there any effort to teach registry and natural history study participants on how to access, understand, utilize, and aggregate data for their own advocacy or for sharing with their own communities? Noah, did you want to maybe start on that, or is that?
So I'm happy to speak a little bit to it. I would say it's a really challenging question to answer generally, since it's going to be something that hopefully is built into how these studies are designed. And maybe the real answer is, making sure that there is a patient advocate, there is a representative from the PAO that has a seat at that table. And from there, I think that's going to be the best channel for communication. One of the things that happens in this space is that each of the stakeholders has a different set of parameters under which they operate, and they have a different set of goals. We talked a little bit about the difference between being published versus some actual clinical endpoints. And I think something very similar happens as study design goes on. So I would say that there is definitely somebody involved in a lot of the studies that are getting created today that can speak to that. I don't know that I can give you a general answer.
Does anybody want to add a little more?
Well, and this is Janine. I can add, the publication that came from the work of CureGM1 with various researchers and patient families that I put the citation on my slide, where I was mentioning there were many bereaved families that were interviewed during that process of collecting the data. Really, that publication was asking the question, what clinical outcomes for any potential treatment are important to the families? And, we see a lot of clinical trials in rare diseases looking at blood biomarkers, or specific, really objective, hardcore scientific markers. And one of the questions there for these families was, what's meaningful to you? Do you want your child to actually improve? Do you want your child who cannot walk or talk to get up and walk and talk and live to be 80 years old? Which would be essentially a cure? Do you want a cure? Or are you content with having slowing of the disease progression? Or do you need at least the disease progression to be stopped? And this was meaningful, and the pharmaceutical companies and the FDA may look at this and say, that's all great, but we really need a great biomarker where we're measuring something, and that's the only thing that will mean this treatment is meaningful, or we actually need to see a cure. But what we are seeing, the landscape changing right now is, and this is coming from patient advocacy group efforts, and I think patient advocacy groups can continue to do this, is we're seeing a change in clinical outcomes that the FDA is accepting in some cases. And we've seen some rare disease drugs come to market, come to commercialization with only, 7 to 12 patients in the trial. And they actually didn't even have the same primary outcome for each one. The physician doing the trial with the patient and the patient both got to vote on what they wanted as a clinical outcome. And maybe there was a grid saying you could vote on these 4 different things. And that was accepted as meaningful, and it wasn't a cure. So I guess I just wanted to add that to this. This is a changing landscape, and I think pooling this data might help us understand that better too, because we really don't have any cures for rare diseases. And for most diseases, we don't have a cure, but we have therapies that can hopefully make patients live longer and have better quality of life. Am I going off track on saying all that, or is that kind of in the direction of your question?
Well, I do believe, it's all very useful information. So thank you. This track is important as well. I do think a little bit actually also might even go back to just, it's easy enough to say, patient advocacy organizations can go into the aggregated data and have a valid request as well. So I think that as a data access requester, that's an option. A patient advocacy organization with a valid scientific question, which is really what's necessary here. We don't just obviously allow opportunities to just take a peek at the data. That's not the way these things work. You need to have a valid research question. So if the patient advocacy organization has that, you're not, at least in these particular situations, going in to snapshot your own data just to take a look. That's not really the way that a lot of this works. The aggregated data has to be put together, deposited somewhere and utilized, and then a question needs to be put to it to understand what's next. But Teresa, you may have been thinking about something slightly different, but that's the typical approach to that. No, did you have something you wanted to add?
Yeah, I was going to say that that's an argument for having both an ongoing natural history study and a little bit less strict patient registry, where you can pull some of these questions and say, hey, at our last 3 Ask a Doc panels, everyone was asking about motility issues. Can we get a GI specialist in here? And the way that translates is, hey, let's go and look at the natural history study and see what age those issues first crop up among our children. Could this be an outcome measure for a potential clinical trial? Could this be something that gets included in the natural history study moving forward? It's just a couple of degrees of translation to get to the right question, which is really, I think, what Kara was saying.
Yes, and actually, that's a super good point. I'm really glad you brought that up. Teresa, in her role, does manage a registry, and they do, I believe, make that kind of interim assessment pretty often. So that's a really great point. Thank you for making that distinction. So I think we only really have time for one more question. Is there anyone in the audience that wants to say something? I can pull another one from here. Okay, all right. We'll just do one question here, which actually is kind of interesting. So the SAEs and AEs that occur just kind of over the natural course of treatment, even non-intervention studies like a natural history study that may have an interventional component to maybe take an MRI or there's some kind of sedation associated, you may end up with AEs and SAEs. the question here was, how does that get reported? How is that information tracked in a non-interventional study like we're talking about here, but where you still might see some outcomes that were not intended? Does anybody want to speak to that? How that actually gets in the data dictionary? Is that something, how is that classified? How is that used? Does anybody have information to speak to that? Okay, well, this is something definitely I think we should think about a little bit, because I do think that that's worthwhile. Janine, go ahead.
To make a quick comment, I'm just thinking about our natural history study.
Mhm.
You know, it goes through the IRB as a non-interventional study, basically, but we do have a section of sort of boilerplate language on what are the MRI risks or, you know, sedation risks, etc. But you raise a really good question. Is I don't think any place in that IRB protocol is there a section that says that we need to report any adverse events that happened if somebody had an adverse reaction to sedation or to, you know, some sort of procedure. So that's a good question. I don't know if other natural history studies have that, but I don't believe ours has an adverse event reporting. I think they sort of looked at it and said, it's non-interventional.
Right.
So we're focusing on that.
I think the closest that it comes to with that is terms of imaging, if there's incidental findings in the MRI, but it doesn't specifically talk about adverse events. You know, incidental findings, we don't even know what some of those are with rare disease. So that's a very good point.
I think Janine gets to the heart of the IRB and what the protocol says to collect. And the data sets that we're collecting are such different data sets. So are we collecting AHR? Are we collecting registry? Are we collecting natural history? Companies have approached us about collecting safety data specifically, AAV-based trials and safety data related to it, because that will really help the future of trials and what have we learned from them. There's a lot of communities that are way far ahead of other communities, and what can we learn from them? So yes, there's an opportunity there, but how like some companies are very willing to share, others are you know, we're working on it, and we would like to have that type of repository at C-Path as well.
Great. Okay. Well, it looks like we have come to the end of this workshop once again. So I want to thank everybody so much for all of our presenters, Noah, Heidi, Janine, and Krista, for all of your hard work putting this information together and for your willingness to have this conversation with us and to share your experiences with our attendees both now and in the future. So thank you very much for everybody's participation, and a special shout out again to Nicole, Olivia, and Leslie, who has been supporting us greatly both at C-Path and the Orphan Disease Center. All of your work is so appreciated, and we look forward to seeing everybody next time. I'm going to go back to my slides here real quick. Hold on one second. Of course, now I can't find it. So hold on one second. Oh, here we are. Okay. So there we go. So thanks again to our presenters. And I did come up with a very quick session summary. This is something that you will see on the website as well, and my slides will include it. But we have a lot of information here from all of our presenters. I'm not going to go everything because we're at the end here, but we're really grateful for all of this feedback. This will be integrated into these papers, and you're welcome to participate as we start to write them. And then next, coming up is our final session number 4. I can't believe it's almost over. So here we are. We actually have all of our presenters at this time, which is very exciting. But we always need more case studies. So please use this feedback form QR code or go to our website and you will find that information there. We need all of your feedback to make sure this is the most useful as it can be for everybody involved. And as many case studies that we can have, even de-identified for your privacy protection if you're not willing or able to share where that sponsorship came from, but you want to share the experience for others that may be looking to share their data. So thanks again, everybody. And we will see you on Monday, June 2nd. Have a great day.